Starting with plasma proteins, because it is what is left over from the last talk. We talked about protein metabolism 1, and in protein metabolism 1, the two topics that I should have covered were plasma proteins, amino acidureas, and protein calorie malnutrition. So this time, I'm going to cover this very briefly to complement what I did the last time on protein metabolism. Then I'll go to my main talk for today, which is the liver's function and liver function test. And I'll give you a slight overview on the effect of drugs on liver's function or on the liver. So here we go. I have these objectives put together for plasma proteins. By the end of this session, I think you'll be able to define plasma proteins, describe their cellular distribution, synthesis, functions, and synthesis and their functions. Apply electrophoretic patterns to classify plasma proteins and discuss clinical significance of the plasma proteins. Plasma proteins are macromolecules which are basic to creation and maintenance of cellular life. They are the final products of genetic coding. So the main building blocks of proteins are amino acids. And the simplest protein is a dipeptide, having only 2 amino acids. The longest of plasma proteins are found in plasma, but can also be found in CSF, pleural space, amniotic fluid. Feces and urine are not part of the body, so we can exclude that from this slide. So plasma proteins are found in plasma, but can also be found in CSF, pleural space, and other serous spaces that we have in the body. What do they do? Plasma proteins can maintain colloid or osmotic pressure, and this is what is called the plasma protein oncotic pressure. It is this pressure that distends and maintains the blood vessels open. So plasma proteins maintain the blood vessels open. They transport materials in blood, and the materials that are transported are the non-water soluble metallic elements and toxic materials like bilirubin. So proteins confer immunity, the plasma proteins. These are the antibodies, and their main function is to combat any invading toxic or noxic materials. They also cause the blood to maintain its fluidity. In other words, they balance between coagulation and fibrinolysis. They also serve as nutrients, and the main nutrient here is albumin, which provides the endothelial cells with their own protein nutrition. Plasma proteins are characterized by the way they move in the electric field. So the electrophoretic mobility is the main method for characterizing plasma proteins. I said proteins, some of them stay in the cells, and this we don't see in plasma. Those that are synthesized on demand are found in circulation. So the ones that are found in circulation are enzymes, and we have some plasma enzymes, and then we have hormones that are transported from areas or regions of synthesis to other places. The most important protein separation technique is plasma protein electrophoresis. So it separates serum proteins or plasma proteins into 5 fractions. The most abundant fraction is albumin, and this is followed by alpha 1, alpha-2, beta, and gamma globulins. In other words, in this separation, we separate plasma proteins into albumins and globulins. And we have albumins and globulins. So this is the electrophoretic picture of plasma, and it gives you to stay very close to the point of application because they are larger proteins. Albumin migrates farthest away from the point of application because it's the smaller of the proteins. Before albumin, there's even a smaller one called pre-albumin. So this is the distribution. This distribution can also be corroborated by densitographic assessment of plasma. A densitogram shows you quantitatively how much protein there is in circulation. Although this electrophoretogram also shows you how much the most abundant of the plasma proteins, as you can see it from the deep concentration of the dye on the albumin section. That means albumin is the largest in concentration. So this is the electrophoretogram. It still shows you the same distribution, and albumin seems to be the most of the plasma proteins. So albumin is commonly preceded by a slight band called prealbumin. So that slight band, prealbumin, resembles albumin, but it has no known function. It is thought that it is used to transport visual proteins or visual pigments in intrauterine life. So it transports the rods and cones to the cornea, or sorry, the retina for their synthesis or for their incorporation in the developing eye. Albumin is the main plasma protein. So anytime we talk of plasma proteins, the first thing that comes to your mind is albumin. It is the one that contributes mostly to maintaining the plasma protein oncotic pressure. It is the one that transports many of the materials we transport, like calcium, bilirubin, hormones like thyroxine, and even drugs. When we take drugs in circulation, albumin is the main transporter. It is also the main nutrient of endothelial cells. So, clinically, albumin is increased. So albumin is decreased in chronic malnutrition and chronic liver disease. So in a clinical setup, when we have malnutrition and chronic liver disease, we have a reduction in albumin. It is mainly synthesized by the liver, and it will be one of the proteins we'll use to assess liver's dysfunction, one of the synthetic proteins for liver's dysfunction. So albumin is occasionally a marker of dehydration. So it's markedly increased in dehydration. The alpha-1 globulin is one which follows immediately after albumin in the electrophoretic migration. It contains 3 proteins, and these 3 proteins are alpha-fetoprotein. Alpha-fetoprotein has no known function, although in fetal life, it is the main protein to maintain fetal circulation. Alpha-fetoprotein has a clinical significance in adult life or in extrauterine life. Its elevation signifies a malignancy in the liver. So hepatocellular carcinoma is a majority of times diagnosed using an elevation in alpha-fetoprotein. So clinical implication of elevated alpha-fetoprotein is hepatocellular malignancy. Alpha-1 protease inhibitor is a watchman of protease function in circulation. So its main function is to inhibit all protease activities that occur in circulation. A majority of cellular organs utilize proteases for their main functions. As soon as those functions are completed, this anti-protease inhibitor comes upon and inhibits the functions of these enzymes. If we do not watch these enzymes, we would have a necessary or inappropriate digestion of tissues. So one indication of lack of this protein is recurrent infections in the lungs of infants or also of children after delivery. So recurrent fetal pneumonias in childhood due to excessive protease excretion in the alveoli, which is not watched. So alpha-1 protease inhibitor protects us from digestion of the hollow organs like the lungs. So emphysema in early childhood is an indication of decreased alpha-1 protease inhibitor. Prothrombin is a clotting factor. It is one which is used, or it is also synthesized by the liver, and it is used to measure or to assess the liver's early dysfunction. Prothrombin, lack of prothrombin is increased bleeding. So a coagulation disorder in which there is excessive bleeding through a prothrombin would be a marker. So clinically, prothrombin is a marker for a bleeding disorder or a coagulation disorder. So that's alpha-1 globulins dose. Okay? Now we go to alpha-2 globulins. And this here we have numerous proteins, and most of them are actually transport proteins. So we have ceruloplasmin, which is a copper transporter. Copper is an important cofactor in the iron metabolizing enzymes. Okay, so copper deficiency leads to improper iron metabolism. Okay, so alpha to this protein confers or transports. So it's transported to hematopoietic centers where hemoglobin is made and where heme requiring iron is incorporated. So its deficiency leads to iron deficiency anemia, and its excess leads to a disease called Wilson's disease. So copper deposition causes hepatolenticular degeneration, which is Wilson's disease. So excess ceruloplasmin, deficiency of ceruloplasmin leads to copper deposition. Increase in copper transporting protein leads to decrease in copper transporting protein leads to an anemia, and an increase in copper transport leads to hepatolenticular regeneration of the liver. So haptoglobulin is the next protein, and this protein binds hemoglobin. Hemoglobin is a main oxygen transporting protein that is synthesized in the reticuloendothelial system. Anytime the red blood cells break down, hemoglobin pours out, and if not watched, that hemoglobin would be excreted, excreted into the renal tubula and excreted. Haptoglobin is a protective protein, so it mops the hemoglobin that is being released from the red blood cells and keeps it for future red blood cells to utilize. Excessive reduction in haptoglobulin in circulation means there is an increase in release of hemoglobin. In other words, it's a marker of hemolysis. Alpha-2 macroglobulin is a large protein whose definite function is not known, but in nephrotic syndrome, when albumin is lost in large quantities, it is increased in circulation to protect or to compensate for plasma protein oncotic pressure. Thyroxine binding globulin transports thyroxine, and thyroxine binding globulin deficiency leads to hypothyroidism. Thyroxine binding globulin in excess means that thyroxine is not adequate in circulation. So hypothyroidism is indicated by increased thyroxine binding globulin. The transcortin is an enzyme, so it's a protein that transports steroid hormones, and we have two steroid hormones that we need to be transported mainly. The steroid hormones that are transported are cortisol and aldosterone. So these steroid hormones are transported to their respective places. So lack of this transcortin leads to reduction in the effects or the effective utilization of those hormones. Okay, the cortisol in reduced gluconeogenesis and aldosterone in reduced sodium retention. The beta fraction contains these proteins. The C-reactive protein is mainly an indicator of inflammation. It has no other function. Its function is to indicate that there is an inflammatory process. So beta-2 microglobulin also has no known function, but it has been shown that it is excreted properly, like creatinine, into the renal tubular system and not reabsorbed. So it means it can be utilized for assessing glomerular function in renal disease. Transferrin is an important protein in transporting iron to its utilization stores. So transferrin increases in, a high level of transferrin in circulation means a deficiency in iron, and a low level of transferrin in circulation means that iron in circulation. Now we get to the one that we use for immunity. This group comprises all antibodies that are body's humoral defense. I have included IgA, and IgA is not a plasma protein. IgA is a secretory protein, and it is found in the alveolar spaces and in the intestines, in the basis for where microbial organisms can invade. Okay? IgD and IgE are also not plasma proteins. They are mainly cellular proteins, and their main function is to modulate immunological reactions. IgD and IgE are all they're found in mast cells, and mast cells degranulate when the IgE or IgDs are engaged in an allergic reaction. IgG is the most abundant circulating antibody, and it is one which combats or which is generated for long-term immunity. And IgM is the main surface antibody in immunoglobulin producing cells, the B cells, and it is the first protein that appears in an inflammatory process. Because it is multivalent, it is a big protein, and it can combat more of the antigen that comes in. The clinical significance of antibodies in circulation are indications of malignancies. One is one in which there is plasma cell malignancy that's leading to excessive antibody production. Like clonal antibody productions occurring in multiple myeloma would be diagnosed using an elevation in these immunoglobulins in circulation. Okay, so in the plasma, in the electrophoretic assessment of plasma, an increment in the gamma region of the electrophoretic pattern is indicative of a malignancy or an excessive immunoglobulin producing malignancy. And one of them is multiple myeloma. Although lymphomas can also do the same, and plasmacytomas can do the same. So IgGs or immunoglobulins are significant in diagnostic assessment of malignancies. So plasma proteins are finished at that end. So you are able now to define them, locate their cellular distribution, synthesis, and functions. And you have identified fractions in plasma that are of clinical significance. So that's the end of my first talk. So